Okay, so this is an introduction and it's physiology, but really when you teach anatomy and physiology, when you learn anatomy and physiology or biology, you learn about how things work, right? So anatomy is really the structure, physiology is more the function. And I don't know how all this starts. It's just crazy how something like a subatomic particle or even a subatomic particle like a quark could actually work. So again, in this chapter, you'll see the organization of life, because this is human physiology we're talking about. Ultimately, we want to know about human physiology. So what are the smallest particles? What are those tiny little things that we can't see?
Atoms.
Even smaller than that, right? Atoms are the smallest particle complex, but what about the subatomic particles? You got to know about these. We'll talk about those in chemistry, but let's introduce this. The subatomic particles. I don't even know if your book really goes into them in this chapter, but what's the positively charged subatomic particle?
Protons.
Yeah, that's a proton. Because this Earth, you know this Earth thing, this round thing, it's round, so I don't want to hear any arguments about the Earth being flat, but we'll talk about that another time. It has two poles. You know, the North Pole, right? Which, you know, they have the big fat guy with the red suit. And then you have the South Pole. So it's polar for a reason. And there's a magnetic field. So when you see positive, there's going to be a negative. And that's going to create energy within that magnetic field. Makes sense? Crazy. So what about the neutral one? That's a neutron. You ever hear that? Neutron? And that has no charge or neutral charge. Now, both of these particles have mass, which means they have a bit of weight. They're pretty equal in weight, actually, depending on how many of them are in there. But if you just have one, then the weight of a neutron is basically the same as the weight of a proton in most cases. Now, what's the negatively charged one?
Electron.
That's the electron, right? Electron. Now, this one is extremely light. This proton and neutron, they like to stay home. They like to stay centered in an atom, right? They kind of just stay there and they make up the mass of the atom. But this guy or gal, the electron, loves to party, loves to get outside of that home, loves to go out. So this is the one that creates energy, that likes to bond. So a lot of things that go on in physiology involve gaining and losing electrons. We're going to talk about in chemistry. We'll talk about ions. Sometimes we'll talk about isotopes. We'll talk about ions a lot, right? So these are the three, and we'll visit them, and we'll talk about how the electron can be shared with other atoms, how the electron can be lost or gained, shared equally or unequally, depending on what the molecule is. So the next one is going to be the atom, with a T, the atom. So I like to say a lot of one atom is an element. So I might, instead of the word atom, sometimes I'll say element, because, you know, we don't use one atom for anything we do in the body. There's no such thing. There's ions, but they come in massive amounts of moles, which you're going to see. So anybody know, love the participation, anybody know what the 4 main atoms in our body are? Name one, just for fun.
Carbon.
Carbon, I would name that first, yeah. Carbon. And by the way, when a molecule has carbon in it, it's most likely that that molecule is going to be organic. That's why they call it organic, not because there's no pesticides or if it's really naturally grown. It's because it contains carbon. And what's the other one? What's the really small one? There's so much of it in our body. It's in water.
Hydrogen.
Yeah, oxygen, hydrogen, right? Hydrogen is the most plentiful element in our body. And then oxygen, you said, right? Anybody know the other one?
Is it nitrogen?
It is. Yeah, so I like to say Chan. That's our body. This is most of the, 97% plus is probably all these atoms, especially the carbon and the hydrogen. So an organic molecule has carbon for sure, and it has hydrogen as well, and oxygen, sometimes nitrogen. So organic molecules we'll talk about in chemistry, because you really need to know about them for physiology. Cool. So the hydrogen, you'll learn, is the simplest. Anybody know what an atomic number is? It's the number of protons within an atom. And usually in hydrogen, non-ionic hydrogen, you have one proton, one neutron, and one electron. So what's the atomic number of hydrogen?
1.
It's just 1. That's the simplest way to do it. So let's learn it now so when we get to chemistry, it makes more sense. Atomic number is 1, which means it only has one proton, which means it only has one electron. So if hydrogen loses that one electron, it becomes positively charged. See how that works? And it becomes the hydrogen ion. I'm not going to go into this deep now, but this is kind of an important ion. I'm going to see this a lot. Like, does anybody know if we have a lot of this hydrogen ion, does our blood get more acidic or basic with more of it? My dog says acidic. What do you think?
Acidic.
Acidic, right. So this is what acid is. Like, you're going to see something like pH is the percent of hydrogen. Now, it's an inverse scale. So the more hydrogen, the lower the number, which we'll talk about when we get to acids and base, hopefully today or next time. So it's percent of hydrogen. So the more hydrogen, the more acidic. Very good. Then you have sodium. I'm not going to talk about the atomic number of sodium, but sodium tends to lose an electron, like hydrogen. So sodium becomes positive. So you've heard of sodium, right? Sodium, sometimes you hear of chloride or chlorine, but if you lose an ion, it becomes chloride. So this, I'm sorry, it gains an electron. This gained an electron to become more negative, a negative ion. So we're going to talk a lot about ions because they got this kind of unstable electron situation going in their valence shell. So they're going to be more likely to bond and cause changes in polarity, especially, remember polarity, like the fat guy in the North Pole and the South Pole, a polar magnetic, creates a lot of energy. So you'll see another one. Do you ever hear of this one? Anybody know what K is? Just K alone without the plus?
Potassium.
Yeah, that's potassium. Very good. Kalima. So that's potassium. So we'll get to know those. And then this is one we're going to talk about a lot with the human body too. This guy loses 2 electrons, or girl. Anybody know what Ca is?
Calcium.
That's calcium, right? Very good. So potassium, sodium, chloride, and there's other things like magnesium, which again could go into its ionic form. And Fe is ferritin, but it's iron, right? Iron. So all these are ions in here. But if you take away the plus or negative, they're going to be stable ions, like just hydrogen without its ionic form, or carbon in its stable form. Hydrogen, oxygen, and nitrogen in their stable form. You don't have a lot of nitrogen ions. You don't want an oxygen ion because then you're talking about free radicals, which we're going to talk about could be dangerous to the body. Okay, so now if we put like 2 atoms together, like say these two, so you have 2 hydrogens and 1 oxygen, or C6H12O6. What do we call those? Or this?
Molecule?
These are molecules. Very good. Molecules. So is water, this is water, right? Everybody probably knows that. H2O. Is that organic?
Yes.
No, it's not.
No?
It doesn't have carbon. But water is miraculous. We're going to talk a bit about water when we go to chemistry, especially when we talk about acids and bases. But know this. Our body is full of fluid. I don't mean like pathologically. I mean normal, physiologically. So water makes up at least 69% of our body. So every fluid, every interstitial, extracellular, intracellular, cytoplasm, the fluid in your ears, the fluid in your eyes, the lymph system, your blood is all made up of at least, 70% water. So totally, put all that together, water makes up a big part of our body. It's a universal solvent, which you'll see. Now you see this molecule right here, C6H12O6. So that's a lot of carbon, a lot of hydrogen, and a lot of oxygen. Is that organic?
Yes.
Yeah, very organic. Does anybody know what that is? It's a carbohydrate. Very good. And carbohydrate, organic molecule like carbohydrate, especially carbohydrate, we want to use for energy. Very good. So might as well talk about it now since we're doing this introductory and get you on the right path. So there's 4 organic, and you will get this in chemistry, but I can't go on without talking about this. And I call these macromolecules because they're big. I shouldn't put that 4 there because it looks like it's number 4, but it's not. So you already told me one of them, and that's a carbohydrate. That particular one, C6H12O6, is actually glucose, which is a monosaccharide. So you have your carbohydrates. Just put carbs. Anybody know another one that has nitrogen? Like if protein. We go to, what is it?
Protein.
Protein, very good. So protein, I know this about protein. Protein is involved in a lot of physiology, especially enzymes. But proteins, we don't want to use them for energy. We want to use carbohydrates first, like glucose. It's definitely glucose. And we'll tell you why. So protein, know this, the protein is the structure of our body. It's our muscles. It's our bones. It's all the connective tissue within our body in the form of collagen in our skin. Protein is our structure. We don't want to break down proteins. Anybody know what the breakdown parts of protein is, or the building blocks of protein is? Those acids.
Amino acids.
Amino acids, yeah. So we need amino acids. We need essential amino acids, and we'll talk about that when we get to the chemistry, right? And we can talk about nutrition and all that too. So it kind of makes sense. Very good. So carbs, energy, protein, structure. What's another one?
Lipids.
Lipids. Lipids is a cool one. So lipids are your fats, your triglycerides, your steroids. Right? Triglycerides, again, we store triglycerides for long-term energy. So it's our storage form of energy, like in the form of triglycerides. But then you have steroids. Anybody hear of steroids? Like what's the anabolic steroid that's released from the testes in the male? It has an "erone" at the end.
Testosterone.
Yeah, testosterone. And the female produces estrogen and progesterone from the ovary. So there's a steroid. It's very anabolic, building things, building proteins. When I say anabolic, what I mean is that proteins are being built. Proteins are being built. And we'll talk about the genetics of all that. So I don't want to spend too much time now, but it just gets so interesting. So what's the 4th one? Everybody forgets this one. Because this is not like we can go to Popeyes or Kentucky Fried Chicken and know that we're eating these. We know we're eating carbs. We're eating the biscuits. We know we're eating protein, the meat on the chicken. We know we're eating lipids because of the triglycerides. But what's the other big one?
Nucleic acids.
You got it, nucleic acids. And you know what DNA is, right? Deoxyribose nucleic acid, and then RNA is ribose nucleic acid. There's a lot of differences between these two, but in the name, it just means deoxy means that there's one less oxygen. Ribose is with the oxygen that it needs. So ribose is a monosaccharide, actually, which we'll learn when we do chemistry, organic molecules. But there's another one I got to talk about. And the other one involves energy, especially the use of glucose and this. Now, this is molecular oxygen, the gas. Like this is CO2, which is a gas as well. So what is another really important nucleic acid that we use for energy? It's our energy currency in every one of our cells.
ATP.
ATP, adenosine triphosphate. You're going to see why that's a nucleic acid, because it has nucleotides, a base, nitrogenous base, a sugar, and a phosphate. So ATP, and I think when it comes to, as far as if you're living, you need ATP. Because if you're not making ATP, you're not living anymore. So you need oxygen to make ATP, obviously. And you should use glucose. I mean, I could use amino acids to make ATP. You know, you've heard of the ketone diet, right? Where you just use lipids, fatty acids from your triglycerides to make ATP. And don't eat carbs. But you're gonna have a problem with that because you're gonna be releasing ketones, right? Ketones could cause acidity in the blood. Are you with me? Crazy. By the way, if talking about acidity of the blood, anybody know what the pH of the blood should be? Like pH is neutral in a laboratory. It's neutral at 7. It's very basic, alkaline at 14. It's very acidic at 0. So a neutral pH in the laboratory is 7. But what should the pH of blood be? Like the one number. I don't want the range. We'll talk about the range later because the range, I'm a little uncomfortable with the range. Because
7.4.
Yeah, I like right on 7.4, right on it. So if you're 7.3, you have acidic blood. If you're 7.5, you have alkaline blood or base blood. Now there is a range, 7.35 to 7.45, but that's a compensatory range. Our blood wants to be at that 7.4. So let's move on before I talk too much. So if you put molecules together, right? And then within a cell, the cell is not the next one. And we're building like complex carbohydrates or polysaccharides. We're building proteins. We're building triglycerides and steroids. We're using DNA and RNA, of course. So what are those little parts inside the cell that has function, like the mitochondria is where oxygen is used to make a lot of ATP through the electron transport chain, or the centrioles are used for mitosis, or the lysosome is used to destroy bacteria. So what's the next one? What are those little cute little organs called?
Organelles.
Yeah, organelles. So for a long time, my favorite organelle is the mitochondria. Now in 7th grade, we call that the powerhouse of the cell, but this is 200 level physiology class. So you'll be telling me why and how it makes ATP and how it wants to use glucose and how it could use other macromolecules like amino acids and triglycerides to make ATP, to give energy. Because if we don't have ATP, or if we don't have oxygen to make the ATP, our neurons, remember those things in my head before? The neurons are what makes up the brain. If our brain doesn't function, or if our brain is not getting oxygen, nothing's going to function. Okay? So we'll talk about the nervous system. We'll talk about the cardiovascular system, which is really important, delivery system, and how the heart muscle is so interesting and unique. Okay, 5 would be the cell. And we're going to live there for a while. I'm going to learn about cellular membrane transport, and we're going to learn about cellular metabolism. We're going to learn about different cells. So what if we put all the cells together? What do we get that? Like one type of cell, we put that together. What are we going to have? Like many of the same cell.
Tissues.
Tissues, right? Tissues. You guys are really good. But we have to go over this. We have to make sure everybody's on board with this. So here's another tough one. What are the 4 basic tissue types?
Epithelial.
Yeah, I always say that one first too. Epithelium, muscle. Thank
Connective.
you. Muscle and connective. You guys are awesome. CT, what was the other one?
Nerve.
Nervous. Wow, this is amazing. Amazing, you guys and gals. So we have all 4 tissue. These are the basic tissue types. So there's 3 types of muscle. We're going to learn every one of them. There's skeletal muscle, there's cardiac muscle, and then there is smooth muscle, which I like to call visceral muscle. Connective tissue is very complicated. It's not just ligaments and tendons. It's not just the stroma that holds organs together. Nervous tissue pretty much controls everything. That's very electric, right? Electric. So now this is very basic. So where would blood go under? That's my toughest question. I'm sure I'll ask it sooner or later.
Connective.
That's connective tissue. Because by definition, connective tissue is cells, like cellular formed elements or cellular structures, and a matrix or a ground substance. So blood, the cells are your formed elements, the red blood cell, which is an erythrocyte, the white blood cell, which is a leukocyte, and the platelet, which is a thrombocyte for clotting. And the liquid matrix is the plasma. So we'll talk a lot about blood, and that's a connective tissue. So bone goes under here as well. it's a calcified matrix with osteocytes. My dog doesn't like those bones. Adipose tissue is triglyceride stored in an adipose site. So that's also connective tissue. And the biggest proteins are connective tissues in much of our structure. You'll hear the term called collagen. In the body, this is the most abundant protein. In the skin, it's keratin, but in the overall, collagen is the most abundant protein. Okay, and the 3 muscle types we'll go through. Those are very specific. Which one's voluntary, which are striated. Tell me about the nucleus. Tell me about the hallmark structures and 3 different muscle types and how you can tell the difference. They all basically contract the same way. Muscle, unfortunately, is not the smartest tissue because it needs an impulse. It needs the nervous system to contract, and that's all it does is shorten to contract to do work. Okay, you with me? Everybody good? Really good so far. So what if we put all 4 tissues types together? What's the next thing we could make in the human body? Organs, yeah, organs. Very good. Organs. So we'll go through the organs when we get to the systems. So 8 would be the organ systems. And we got to cover a lot of those this semester. One semester all this stuff. I've been here for 2 hours. I'm still on these 8 and 9 things. So organ systems, not systemics. And 9 is us, the organism. So, you know, we're animal kingdom. We're mammals, right? We have fur, we have teeth, we have nipples, we have our young carried in our uterus, right? So we could use other mammals for not clinical trials, but research, right? To see if hydroxychloroquine or whatever COVID-19 medicine will work in a rat or a mouse. And then we'll do clinical trials and we'll talk about the scientific method in this chapter, which is really very well done in your book. So everybody okay? So usually when I turn off the share, where you can turn your mics on and take a breath and see how you feel about everything. Everything okay? The basics. So you have some things. You have some memorization that you should go into before you start your readings and start going over these chapters. So it's really good. Let me see what's in the chat. You guys got good stuff. Creatine, that's an interesting one. We'll talk about that. Creatine phosphate is as an energy source, and creatinine is a byproduct. I didn't really talk much about it because it's going to be such a big part of the semester. I mentioned ATP, of course. Remember ATP is a nucleic acid, but I kind of said oxygen and carbon dioxide. So of course we want oxygen to get into our body. We want to get rid of the carbon dioxide. We're going to see how carbon dioxide is a waste product, really. We don't like a lot of carbon dioxide in our blood. It makes it acidic. It causes disease, right? Visceral fat is connective tissue, yeah, because visceral fat is like an insulator for your organs. It's behind your eyes. It's around your heart. Normally, not a big obese BMI above 30 type of adipose. I'm talking about normal adipose. And it's an insulator. It's a storage of energy, and it also releases hormones. And hormones that we don't even know about what they're really going to do eventually. So we're learning a lot more of that. Okay. So again, we'll go into the chapter and we'll talk about each thing individually. But I really think that's real important. So there's one more word I want to put on here when you're talking about physiology, especially, which is the function. What's a term that's used to tell you if the body's in balance or equilibrium, or if all your systems are within range, like your blood glucose is in range, your body temperature. Yeah, homeostasis. Yeah, you're going to say this a lot. So it's a balance, yes. It's equilibrium, yes, but it's also the body's way of correcting something that goes out of range. Like your book talks about it a lot, because I like to talk about it too, is like blood glucose, which Every book is different, but it should be 90 to 110 milligrams per deciliter of blood. So, of course, when we go to Dairy Queen, it's going to go up to 200. And then, but your body's going to release hormones like insulin, especially, that's going to get it back into range, but it doesn't want to overcorrect it. You don't want your blood glucose dropping below 60 and even 90 or something. So it doesn't overcorrect. It keeps it within range, and it responds to change. Like when you eat, when you're stressed out, when you're scared, when you're happy, when you're sick, when your blood is acidic, when you have a virus, when you have a bacterial infection, parasitic infection, or maybe you don't even know what's wrong. All right, so many things can go wrong. So if you go out of homeostasis, that's when you're at risk for disease. And of course, medicine these days, they're treating it old-fashioned way many times, with, okay, let's wait to see what's wrong. And then your PH, you listen to this. Let's wait to see what's wrong, and let's treat it the way we've been treating it for the last 100 years. Does that sound familiar? If right away we'll go to the book, we'll take a look at it. But, everybody's different. Everybody has, remember DNA? We talked about DNA. DNA has been handed down to me for the last 140,000 years or more. So there's genes that are made up of your DNA and your chromosomes. And that's your genetic profile. And it's really important that you get treated the way that your genetics are geared to be treated. So it might not work for you. It doesn't matter if there was 30,000 people in a clinical trial. So medicine now has to be a little more heads up, because we know more about the genetic profile. We know more about genetic mapping. So homeostasis is going to take on a new meaning when you guys are into practice, or you're out of school. So again, the old way we've been doing it really has to change, and that's really important in medicine. That the egos are put aside, and we get the right treatment for the right person. So it's going to be more individualistic medicine. It's going to be more about diet and, of course, exercise, and not over-prescribing the protocol. But for now, that's the best we got. Like, again, if we look back 100 years ago, we'd be laughing our asses off about what they did. And say, that's crazy, but it's going to happen 50 years from now for what we're doing now. Everybody kind of understand that? So when you're taking this class, you should keep that in mind about how physiology works. What's the normal? What's the normal for me in my genetic profile, my genetic makeup? So homeostasis is a big deal, right? Okay, so back to physiology now. I think you got a good idea of what's going to happen here. It's biological function. Biology is the study of life. So in here somewhere is going to be the characteristics of life. So one of the big characteristics of life is being able to respond to stimulus, to change, to create that homeostasis. As mammals too, we want to reproduce too. We want to reproduce not because it's fun on a Friday night to try to reproduce, but because we want to get our DNA down to the next generation and so forth, so on, so we can survive as a species. Right? So there's different qualities of life studied through biology, or the characteristics of life. And we have to be able to repair. We break our leg, it repairs itself as best it can, but we try to help it, right? So repair and growth, all characteristics of life, how the body works. So we have to know the normal physiology of the cells. And there's trillions of cells, but we're going to narrow it down. You saw the organ systems, you saw the cells, you saw the CHON, right? CHON, C-H-O-N, that make up the molecules. And we'll talk about different cells in different tissue, like muscle cells, nerve cells, which are neurons, all the different connective tissue cells. And of course, epithelial cells are pretty straightforward. Epithelial cells, they're probably the classic cell with the cytoplasm and the membrane. And then, the mechanism of action, right? Because if something goes wrong, let's say something goes wrong with the mechanism of action, like this whole thing with insulin being secreted from your beta cells of the islets of Langerhans and your pancreas. That's a normal mechanism to lower your blood glucose when it goes too high homeostatically. So if something goes wrong with that mechanism, you have what's called pathophysiology, which you'll read about. So something's wrong that's going to get me out of homeostatic, out of homeostasis, because the normal mechanism went wrong. And sometimes we need medication. I'm not going to downplay any medications because really that saves our lives. So you have to know the mechanism of action of a medication or homeostatic mechanism that helps change that pathophysiology, like a medication. Like it's easy when you talk about blood sugar. If you're diabetic mellitus, diabetes mellitus type 1, and the problem is in DM type 1 is that your pancreas doesn't make insulin. Maybe it's an autoimmune condition. Maybe it's just straight up genetic that's been handed down to you, this inability for the islets of Langerhans beta cells to make insulin. So what do? This is very simple. And imagine this, at the turn of the century when they discovered this medication. It's just insulin, synthetic insulin. They'll just give you insulin to type 1 diabetics. And there could be diabetics in this class, type 1. And most of us know somebody who's diabetic if we are not, unfortunately. It's a horrible condition. We don't want our blood glucose going out of control. Our kidney will be destroyed. Our blood vessels will be destroyed. And it's not a good thing. It's a comorbidity for other things. So if you get COVID-19 and you're diabetic, you have to be extra careful, especially if you have a respiratory condition, which would be another comorbidity, and being overweight and having a heart condition, atherosclerosis, bad diet, all comorbidities. Okay? And then you explain these things using the cause and effect, like I just said, with something as simple as insulin. Now, there's a lot more detailed things that go on in cell membranes with those ions. Remember those ions? Especially like calcium in the heart. Really important. And of course, we learned all this from good scientific experiments through the scientific method, which your book goes through very, very well. So I mentioned pathophysiology, and that's the concern of a problem with the mechanisms of normal physiology and it leads to disease. So understand the normal processes. I mentioned before that we're mammals, so we try to compare something like a mus musculus, which is a mouse, who, of course, they have a much shorter lifespan. And you may get a lot of arguments from people who study things like cancer cells and leukemia that the mouse model is not really working for us. Because, again, do we have a cure for most cancers? Not really. Especially something horrible like acute myeloid leukemia, which is a very bad condition where you have cancer of your white blood cells that affects, really comes from your bone marrow. And sometimes it's genetic, sometimes it's just bad luck. And why does that one cell create that? So at least we're trying with these other mammals like mice and rats to give them the genes that maybe would cause that. They would give them the actual condition, and we could learn from it. It has worked in some cases, but it doesn't always work. Okay, and this is how we develop drugs. And we know this is big money. We know that there's a lot of pressure. You've seen this in the news lately with, let's get this vaccination out. Let's get a cure out as fast as we can. So we got to test it on animals first, and then test it on humans, and then test it on a lot of humans who actually have the condition, who sign up for this. So you understand how the physiology is discovered, which is really, really interesting. Scientific method this has been around. It's very controlled. It's refereed. This has to be watched. So you make observations like, high blood glucose. This person is extremely exhausted. They're urinating all the time. They're always hungry, but they're losing weight. An observation like that on something, or something you see in the laboratory. Because we're not working in the laboratory, but these things, but you might get to see something like the scientific method being applied towards your labs. So a hypothesis is what you think of. Okay, diabetes mellitus type 1 is high blood sugar that's causing all this. This is it. We think there might be a problem with the pancreas secreting insulin. That's my hypothesis, and that was their hypothesis. So you have to be able to test it, right? And then conduct experiments. Okay, and then you experiment, and a lot is lost. You do these experiments, and you could be wrong a lot of times, and you could fail many times. You could fail for decades before you really find an answer to what your hypothesis is leading you to test. So you really have to put the ego aside and accept it. You don't want to push your hypothesis. I'm right, Nobody else is right. I'm sure this is going to work out. So I'm going to push these experiments to work. And you can't do that. You have to take the ones that go wrong and move on. So you analyze this, and you have to know about statistics and data. This is a whole statistician's dream, right? And then you get all the results, and you need help on this. You can't decide this yourself. You need peer review. You need another scientist, another PhD, another doctor, MD, DO, PA, whatever, to come in and talk about more empirical results and what we see in the real world, right? And then you verify that hypothesis. And then if your peers agree, and the people who referee medical journals like Nature and Science and the New England Journal of Medicine and JAMA, then it could be published as a legitimate conclusion and that came from a legitimate hypothesis. It kind of makes sense. So again, this has to go through, and then we have to test it. If it's a medication, like, or a vaccination, we have to test it. And clinical trials are basically people who either have the condition that they're trying to treat or they don't. So if they don't have the condition, like you're not diabetic or you don't have COVID-19, that's called the control group. And you can set your own parameters for control group of the ages, and depends on what you're testing. So the control group usually doesn't have the issue you're trying to prove the hypothesis of. So here's this is good to know, the research that what's required for this research. You need quantifiable, which means you have to be able to measure in a quantity, compare it to other quantities within that same hypothesis or a similar idea, like vaccinations versus medications versus any treatments or any conditions. Experimental group is usually the one who has the condition, like COVID-19, and the control group does not. So let's see if there's a difference in the side effects of what goes on. Okay, and it doesn't have to be COVID, it could be something else. Testing a blood pressure medication, because that's going to make a physiological change. So people sign up for this. So these are your, again, clinical studies. And then you review the statistics again, and then you can review it by these refereed peer journals. And again, you have to be prepared for some type of negative outcome. So if it's wrong, it's wrong. You start again. And a lot of medications are found by accident. A lot of medications or chemicals that we use are found in nature. Like Viagra, right? Viagra is a drug used for erectile dysfunction. It's really about blood flow. And we're going to talk about vasoconstriction, vasodilation, how it controls blood pressure, your blood vessels constricting, your blood vessels dilating, and how that affects your systemic blood pressure, which we'll talk a lot about. So Viagra was really under research for high blood pressure, but it did more, obviously. And they found a new chemical and they went crazy. Big money, So again, you have to know the economic part of this too when you're studying this stuff. So again, pharmaceuticals is probably the easiest thing to look at. And again, we do the research on the mice, on the rats, and that could go on for a decade before. And this could be also a vaccination could work this way too, because technically that's a pharmaceutical. And then you go into clinical trials, and they start out small, and they should be big. I think like the NIH says that for a clinical trial to really be valid, you need at least 30,000 people to be tested and proven and documented and reviewed through refereeing. Okay, so again, mice, rats, mammals like us. In vivo means in real life. In vitro means in the laboratory. So it's a living creature. Again, they're not the same. Their brains are similar, but the 99% the same as us genetically, but that 1% is a huge difference between the Homo sapiens and the mus musculus, which is the mouse. Okay, so we do a lot of research there. And the points are valid, but not for everything. So again, that has to be understood. So you do the trials, you go on, you do clinical trials, and that's on people, volunteers. So they change the rates of the dosages, and there's a control group, an experimental group. So right now we know there's a hypothesis, we know there's testing and experiments, we know there's an experimental group and a control group, and you know about the clinical trials, and very big on quantitative statistics. Okay. And then we can go on phase 4. Phase 3 is the clinical trials on a large number of people. That, like I said, I think that should be around 30,000 people. And look, and this is really important. This is really important right here, that you include now both sexes, age groups, ethnicities, and people with comorbidities. Because to be honest with you, as horrible as this was, and it was a nightmare, back in the 60s, there was a study for heart disease called the Framingham study for heart disease in Massachusetts. So in the 60s, they weren't testing women. They weren't testing people of color. They were just testing white guys in a certain age group. And this was completely invaluable. I mean, it's valuable for what they learned about atherosclerosis and the stent and bypass surgeries all came out of this internationally. But they forgot to realize that women can suffer from heart disease and their symptoms are different than men. And heart disease may affect other populations more than another. So it depends on where the stork dropped you in the world. If it was dropped in Norway versus I was dropped in Sudan, I was dropped in Venezuela or Antarctica. So it's different genetically because, again, that DNA, by the way, DNA, deoxyribonucleic acid, is only in your nucleus. It never leaves. RNA is the one that leaves and builds all those proteins that makes you who you are. So it varies, and these genes go back for years. So, and evolution, whether you believe it or not, is a process, and there is a natural selection, and this has to be taken into account, right? With evolution. Now, I don't think, and you're probably going to agree with me, that all these diseases we have, especially high blood pressure and diabetes, both of them we have no cure for. We can only manage them. Cancer, we have no cure for it, but we can treat it as the cases come up, which is kind of barbaric and archaic at this point. So we should be able to learn how to treat these conditions as an individual genetically, and we're getting there. So you're going to see, like, why do I have 7 bones in my ankle? Why do I have 8 bones in my wrist? There's really no reason for that. It just causes more problems. Why is my knee designed the way it is? My shoulders are really bad design. And why does my spinal cord stop at L1? And then we have all these other horse's tail at the bottom. Things like that. It doesn't make sense. Why does my brachiocephalic trunk only on one side? And why do I only have 2 lobes of lungs on the left and 3 lobes on the right? So in some cases, evolution hasn't caught up to where we are now. Because I'll tell you this, physiologically, there's 95% more mechanisms in our body that are designed to elevate blood pressure than they are to lower blood pressure. And what's a big problem in the United States, besides heart disease and cancer? It's hypertension, high blood pressure. Because we don't have the mechanisms to lower it naturally. That's where we're taking beta blockers, blocking the amount of calcium that can get into my cells, especially arteries, because the arteries are going to contract and constrict and raise my blood pressure. So you have to understand we're at a disadvantage evolution-wise, if you will. And if you don't agree with me, that's great to tell me. But, it's still, why do we have all these conditions, right? So clinical trials should be very unbiased. Okay? How are we doing so far? How long have we been talking here? Yeah, what do you think? We've been talking too long? Should we take a break yet? Where do we start, 2:30? Let's talk for another 15 minutes, maybe.
I think we're good.
We're here till like 5:21. So anybody have any questions about the scientific method? It's pretty straightforward. It works, but you got to take everything with a grain of salt. You MDs, you PAs, and even you dancers and musicians, you got to take everything with a grain of salt. If somebody tells you something that MRI is the only way you could diagnose this herniated disc in my back or brain tumor in my brain, you got to take everything with a grain of salt. Chris, yeah, you have a question?
Yeah, if you're do you have to be like have a certain credential in order to form an official hypothesis? Like, let's say you come up, you're making
Good question.
you don't have like a degree or anything like
Yeah,
that.
it's funny. I just read something on that. I forget what it was, though. I'm so sorry. It was something pretty important. And the hypothesis and the actual design of it was made with somebody who just had a BS. And it was credentialed, but he would need a PhD to set up and get the grant money. So if you say you are, you want to do something like you're in dance and you want to learn about this iontophoresis of the calcaneal tendon, and you found that this will be work if we use this supplement, and you couldn't just go out and do that yourself if you're in college or you're a TA. Even I've heard stories of TAs coming up with these things, and if you read a lot of the literature, a lot of these ideas come out of graduate students, people going for their PhD or their MS in science. So, but they can't get the money to do these trials and get the laboratory time unless you have a PhD and you're tenured track within a university in the world, like McGill University in Montreal or NYU or Columbia University. You know what I mean? So you would have to be sponsored and be directed by an upper level guy who's probably not going to be that happy unless he's your dad. Anybody have any other questions along the scientific method or the basics of physiology? So far, so good. I think you're all awake. I can't see anybody, so I'm awake. That's all that matters. So here we go. We've heard of this word before, homeostasis, right? And I like the words, balance, equilibrium, consistent environment, but it's really a response to a change, keeping it within a normal range, keeping the disease away. So it works through a feedback mechanism. Let's see where we're going here. Maybe I could put this out. I'm not going to ask you any of this, but this is all interesting. Maybe you get on Jeopardy and you have to answer questions like this about, going back to early in time with Aristotle. William Harvey, that's a cool thing, how the heart pumps through a closed system. Again, what year was that? So that wasn't even, that's a few hundred years ago, but still, technically that's not that long ago. And it wasn't that long ago when other things changed. So Bernard, 1813, something, so he was on the right path, Claude. But then Cannon, he just coined the term. And really, this is where it took off from there. So this was, not quite 100 years ago. But we should be better by now. Like Wally here, he could have really helped us. If he was around today, he probably would have thought more out of the box and worried less about money. And probably would have had better answers to these homeostatic problems. Consistency or constancy, I like it. I like it. Internal, yeah. Blood pH, 7.4, right? That's one. What's the body temperature? Like we're getting that taken a lot. Like if you walk into your school, you got to put your head like forward on our school. What's the body temperature in Fahrenheit?
98.6.
Yeah, 98.6, which is about 37 degrees Celsius, we say. Centigrade, so a metric system. Metric system plays a big part in physiology. I don't know if we're going to go into it, but we'll see. Blood glucose, I think we could say 90 to 110. You should always put the units, whether it's milligrams per deciliter or cc's or something like that. What about the average blood pressure? We're going to talk about this a lot later, but let's just throw a number out there. Systolic over diastolic. Looks like an average textbook blood pressure.
120/80.
It is. I don't like the 80 though. I think it should be lower. But this is, again, Framingham over the years. This is still the average in the United States. What about heart rate? What's the range of heart rate? And this is millimeters of mercury.
60 to 100.
Yeah, 60 to 100. I like that. Ventricular heart rate between 60 and 100 beats per minute at rest. Nice homeostatic range. And I'm sorry, I don't remember the bicarbonate and the CO2 levels, but CO2 is about 35 on the end, and Bicarbonate is like 24 in the end. So you have to know, I'm just going to write these down then for now. I'll do when we get there, like CO2 levels, oxygen levels. I think it's in this PowerPoint actually, it's sort of in the book anyway. CO2 levels, oxygen levels, very important. And bicarbonate levels, this is a buffer, especially for carbon dioxide, this bicarbonate ion. And then of course you have sodium levels, which are really important. This is a really important ion. You have potassium levels. Anybody think of anything else? Like when you go to the doctor, what are some other things we could check for besides the vitals, like temperature, blood pressure, heart rate? You can check respiratory rate, which I believe is like 15 to 22. Breath per minute, not beats. What else? What else? We take a blood test. Let's do a blood test. We can get carbon dioxide from an arterial blood gas. You can get oxygen, overall oxygen. You can get bicarbonate levels from a blood gas and a blood draw. You can get, what is it?
White blood cell count.
Yeah, your blood count, your CBC.
Plasma level.
What was it?
Plasma level, the level of fluid.
Yeah, the level of plasma and hematocrit, like how much red blood cells, and hemoglobin, which you'll learn a lot about, and the plasma to see if you're dehydrated. The blood volume, very good. That's a good one. So we need to talk about that in physiology. And then you have your lipid panels, right? Your LDLs, your HDLs, your triglycerides, total cholesterol should be within homeostatic range. Also, CBC is important, for your immune system and your red blood cell count, and which is white blood cell count and platelet count. But there's a condition, not a condition, but a cause, a change. It's a normal mechanism. It's an appropriate mechanism, but it can cause more problems. Like, did you ever hear of autoimmune condition? Like an autoimmune condition, like myasthenia gravis or multiple sclerosis, maybe even diabetes type 1, could be an autoimmune condition. Now, what does that mean? Anybody know? Anybody want to talk before we take a break?
I'm actually a type 1 diabetic.
Uh-huh.
So an autoimmune disease is like when your body attacks itself and something stops working as a result of that attack.
Yeah, we're going to have to talk a lot about that, autoimmune. So it's your white blood cells, basically, your lymphocytes, and sometimes your neutrophils and eosinophils attacking your own tissue, attacking your own cells, your own proteins, which could be receptors, could be enzymes, and even epithelial cells that are glandular, producing hormones, like in this case. So autoimmunity, and we don't know why. A lot of times we don't know why, and we have to figure it out. We have to know why certain populations. Why does MS happen more in females? Why does diabetes start so young? Diabetes type 1 starts so young. So it's your own white blood cells, which we can see in a CBC or a SED rate, which you'll see, attacking your own cells, like your receptors, which I'll get into. But just getting sick, just having a condition like a viral infection or a bacterial infection, it's completely appropriate for these white blood cells to be proliferating and doing their job, destroying what they need to destroy. But when it happens to normal cells, we got a problem. So there's a term. Anybody know the term for like excessive like macrophages and histamines being released in your system? Like when you get stung by a bee, or when you sprain your ankle, or when you have a vascular headache. What's happening to your cells and tissue? What's that word? Begins with an I.
Inflammation.
Inflammation. Very good. Now, inflammation is a completely appropriate response when it's necessary. It's important. It causes, blood to clot a little easier if you're injured or you have surgery. It's completely appropriate. But what happens is you have too much of this, too much inflammatory cell release, which you're going to learn. And this can cause a lot of problems. First of all, an autoimmune condition, possibly, but you're going to get things like heart disease, or of course arthritis, right? You've heard of that. Arthro means joint. And there's autoimmune immune types of arthritis, like rheumatoid arthritis or psoriatic arthritis or gouty arthritis. These are all results of inflammation. Inflammation can affect your blood vessels. It kind of destroys the walls, the inner walls of your blood vessels, pure and simple, right? So generally, here's a good question. Inflammation. Let's talk about this now. What the hell? Before we go into these feedback loops. It's a law class. So let's talk about inflammation. So I have a headache, or my back hurts. And we don't know, but it could be due to some inflammation. What would you take?
Aspirin.
Ibuprofen.
Yeah, perfectly said. Yeah, ibuprofen is like Advil or Motrin. Aspirin is acetylsalicylic acid. So these drugs are anti-inflammatories, non-steroidal anti-inflammatories called NSAIDs. Now, a steroid would be something called cortisone, if you heard of that. That's a steroid anti-inflammatory. So our body releases cortisol when we have inflammation. So that kind of reverses the mechanism. But remember this, pretty much any drug you take, including those non-steroidal anti-inflammatories that you mentioned, is going to have some type of side effect. So you can't just treat that one vessel. You can't just treat that one bone in your back or that one joint in your back. So you kind of follow that? So they have to be controlled, and they're not perfect either, but they do work. They decrease inflammation. I've yet to find a doctor who doesn't agree that taking a low dose aspirin every day is beneficial for you, whether as an anticoagulant to prevent strokes and prevent myocardial infarctions, which are heart attacks, prevent embolisms, blood clots of other natures, deep vein thrombosis, but also to prevent Alzheimer's, to stop the inflammation in your brain that could destroy your connections between your neurons and your synapses. So again, why I brought that up here, I don't know, but I think it's really important. So homeostasis is accomplished by feedback loops, mostly negative. I think all the feedback loops, except for maybe 3 that I can mention right now, are negative feedback loops. And what does that mean, negative feedback loop? A negative feedback loop means, like, give me an idea. Give me a one of those homeostatic things that you get at the doctor, besides where you don't have to draw blood. Like body temperature, or blood pressure. Yeah, let's talk about blood pressure, because that's a little more physiologically. So the stimulus is raising blood pressure. Let's talk about blood pressure raising, right? Blood pressure raising out of homeostatic range. So let's say above 140 over 90. Our blood pressure, and this is at rest. Of course, our blood pressure is going to go up when we're excited. Our blood pressure is going to go up when we exercise. All normal. But we got to talk about this is at rest. This is just sitting in your house, breathing normally, not being excited. Your blood pressure, you just can't get it to lower. Well, you don't know, actually. When you have high blood pressure, sometimes you don't even know it. So there's nothing you could do unless you want to meditate. But when your blood pressure goes, let's say over 150 systolic, which is high blood pressure, you have mechanisms in your body that are shut off, basically. Like, you'll probably urinate more. So your kidneys will get rid of more water to lower your blood volume. Like somebody mentioned plasma before, and that's important. So you're going to pee more. You're going to decrease your blood volume. And in the same time, hormones are going to be released that get rid of sodium and water at the same time. So we have what's called diuretic hormone, or anti-diuretic hormones is shut off. So it's going to cause your blood pressure to hopefully go down by decreasing your blood volume by urinating more. Now, the nervous system, of course, we're going to talk a lot about this, and we should mention it now, of course. You have what's called the autonomic nervous system, which has 2 branches. Anybody know what those 2 branches are? One's fight or flight, the other is rest and digest, if that helps you.
Sympathetic and parasympathetic.
Righto, Claire. Dr. Claire, you're on it. So, so sympathetic is fight or flight. So that's going to increase everything, heart rate and blood pressure, and also hold on to more water. So your sympathetic nervous system, if your blood pressure goes high, has to be depressed or inhibited. So that would be parasympathetic, would take over, hopefully. But unfortunately, it doesn't really affect your blood vessels, but it affects your heart. So those are the mechanisms. But I told you before, if you heard this, what I said, so important for later on, our body has much more mechanisms to raise blood pressure than it does to lower them. In fact, I can only name one hormone that's released from your heart that actually lowers blood pressure and gets rid of sodium, and gets rid of water, and turns off your antidiuretic hormone. So the point is, the negative part is, it's not going to lower your blood pressure so much that it goes below 50. It's going to, the output will be shut off. And we're going to see the parts of the actual feedback mechanism. So the initial output is shut off, where we're not going to keep lowering the blood pressure until the person is in anaphylactic shock. Okay. So the first thing we need is a sensor, and this is detecting change. So let's not mess around. This is called a receptor, and this is nerve tissue. So the receptor for blood pressure. Now again, we'll learn this when we get there. Let's just use blood pressure because you brought it up. The receptor for blood pressure are called baroreceptors, as baro means pressure. And they're in very large blood vessels, your carotid or sinus and your aortic arch above your heart, left ventricle. So they sense blood pressure. So if they're getting bombarded, they know the pressure is good, so they're not going to send sympathetic signals to make the blood pressure higher. But if it's too low, they'll send a signal to raise the blood pressure, but trying not to raise it above, so that's a negative feedback. So the receptor is what senses the change. Then you have an integrating center, which is really a control center. And most of the time, the control center is somewhere in your brain. Not the brain we think about, not our cerebral cortex, but places like the brainstem, the medulla oblongata, or the diencephalon, the hypothalamus. These are your integrating centers or control centers. So the input, you have input, well, actually you have stimulus, I'm sorry, the change of stimulus. So the stimulus is actually a change in whatever condition we're going to talk about. And then you have input to the brain or control center. I'm sorry, input. So in the case of blood sugar, glucose in the pancreas, you don't need the brain. The pancreas is the control center. But most of the time, the control center is the brain. Most of the time. Not every single time you're going to see it. Those parts of the brain that are deep brain that we don't think about. So they're going to send out an output. The control center is going to send an output. Sympathetic or parasympathetic or a hormone or insulin versus glucagon, which is like insulin's evil cousin made from the alpha cells in the pancreas. Maybe it could be calcium homeostasis, where certain hormones elevate blood calcium, certain hormones decrease blood calcium. So the output is either a hormone, a nerve output, sympathetic, sometimes somatic, which we'll learn. Now this is the hardest thing to remember. Effector is the part of the body that's working, that's doing the change, like your heart. And you'll learn about the heart conduction system, the sinoatrial node and the atrioventricular node. It could be your blood vessels, it could be a gland, right, like a sweat gland to release water. Right? It could be other muscle, like skeletal muscle. So they produce the change. So they're the effectors. So if we want the heart to increase heart rate, if heart rate is the issue, then the SA node of the heart is going to increase the heart rate, increase the action potentials, which we'll learn. If we want it, and then of course it's not going to increase it so much that it goes out of homeostatic range, like you told me before, it was 60 to 100 at rest, ventricular heart rate. So if it goes above 100, that's tachycardia. You don't want to lower it so much below 60, like to 40 or 30, where it's bradycardia, because that is not consistent for good cardiac output. Same thing with the blood vessels and blood pressure. The glands for releasing hormones. Skeletal muscle for contraction, like shivering when you're cold. right? So we'll learn about skeletal muscle, but in this case, it can be an effector, the organ or the gland or the muscle that's actually doing the work to make the change. We don't want that to go over or out of homeostatic range, so it has a shutoff. And the shutoff really comes from the control center. That'll shut off the output, and that's negative feedback. If it was positive feedback, that output would be enhanced, and the heart rate would go out of control. Your blood pressure would go either drop really bad or go up really high. You'd produce too much insulin, and you'd have no blood sugar. You'd have extremely low blood sugar below 90 milligrams per deciliter. And you don't want to continue to shiver until you go into spasm, and you produce so much heat that your body temperature goes above 102. So it has to be shut off. Positive feedback, it's going to go up and up. So most are negative feedbacks, and we'll talk about the positive feedbacks. So most are negative, and it moves in the opposite direction of the change. It stops the output, so it doesn't go out of homeostasis. My internet connection is unstable. Okay, so you make a change at a set point, and the set point is set by your homeostatic mechanisms that are built into our body and mostly controlled by the brain. Okay, and it's a continuous process. It's happening right now. We sat here, homeostasis is going crazy in our bodies right now. You know, we're curing cancer in our body. We're fighting viruses and bacteria in our bodies right now. And we're also maintaining our blood pressure and our calcium levels and our blood volume and our glucose levels and so on and so forth. So this is just a schematic about keeping things within range over time and not deviating. Again, more body temperature, we kind of talked about that. So 37 degrees Celsius is in the middle. So it has a little bit of leeway. You know, you've probably seen that when you get your temperature taken a lot lately, when you go into restaurants or you go into buildings or get your nails done, whatever, they take your body temperature. And it could vary by a couple of points. Very, very, not a big variance though, for blood temperature. So your effector, like I told you before, if you're hot, is going to be the sweat glands to release the water, because water carries a lot of heat in it, and when it's released, it takes away some of the heat. Okay, so we have the sensors, integrating sensors. In this case, it happens to be the hypothalamus, that's a thermostat for body temperature. And the end product is, once it starts to go out of homeostatic range, it shuts off. So the integrating center shuts off the output. Okay, so maybe we'll just take a break now. We've been here a long time. Everybody agree? We take a break?